Hello viewers, this is Ramakrishnan, Assistant Professor, Department of Biochemistry, St. Joseph's College of Arts and Science, Cuddalore. In today's program, I am going to discuss the topic mutarotation. It's the phenomenon of change in the optical rotation of an optically active compound with respect to the time and reaches to a constant value after a period of time. Generally, this character is observed in carbohydrates, which is having the reducing property. This term was first coined by French chemist Augustin Pierre Dubrunfaut in 1844, and he has also discovered the structure and characters of sucrose, fructose, and maltose. In order to understand the concept mutarotation, we should have some idea regarding plane polarized light, chirality, ionomers, optical activity, and polarimeter. First, plane polarized light. When we consider a light source, it will emit the light, but the emitted light is a normal light and it will propagate in all directions. When we pass this normal light through a Nicol prism, it converts the normal light into the plane polarized light. That light will propagate in single direction, and we are going to use this light for measuring the optical activity of a compound. So next, chirality or chiral carbon. Generally, organic molecules containing carbon and the carbon four valencies are satisfied by different atoms or groups, then those carbons are called as chiral carbons. For example, carbon A, the carbon which contains A, B, C and D in four valencies are called chiral carbons. Next, anomers. Anomers are a pair of isomers having the same molecular formula, structural formula, etc., but differ in the spatial arrangement of atoms or groups in a single asymmetric carbon which is formed because of cyclization or anomerization. That carbon is called anomeric carbon. In our structure, we have the example of alpha D glucopyranose and beta D glucopyranose. This pair is an example for anomer. When we look at the structure of these two, in carbon 1, alpha D glucopyranose have hydroxyl group in below the plane, whereas beta sugar will contains above the plane according to the Haworth projection formula. These two pairs are the best example for anomers. Next, optical activity and polarimeter. Optical activity is the tendency of a compound which will rotate the plane polarized light towards either right direction or left side direction. So a mechanical part or an instrument used to measure the optical activity is called polarimeter. Commonly, polarimeter will contain a light source and a polarizer, the unit optical device used to convert the normal light into a plane polarized light. When this plane polarized light is passed through a solution such as glucose, the emerged light will be rotated. If the rotation is in the right side direction or clockwise, then those compounds are called dextrorotatory compounds. And if the light is turned towards the left side, then those compounds are called levorotatory compounds. So the dextrorotation and levorotations are all explained or represented by the plus symbol and minus symbol. This is generally observed in the chemical bottles. So a chemical contains plus symbol means that compound is dextrorotatory, and similarly minus symbol means levorotatory. So come to the point. So mutarotation is the change in the specific rotation of an optically active compound with respect to the time. So this is time based one. So in order to explain this concept, we are going to consider the example glucose. So when glucose is dissolved in water and placed in a polarimeter, we will observe the following changes. If the glucose is D form, pure D pyranose, then it will exhibit the specific rotation of plus 112.2 degrees. That means dexorotatory compound. Then after a period of time, this 112.2 gets gradually decreases and reaches to a constant value of 52.5 degrees. And similarly, when you take a beta D glucopyranose form, that will have the tendency of rotating the plane polarized line towards plus 18.7 degrees dexorotation. And when we place it for some times, this 18.7 gradually increases and reaches to a constant value of plus 52.5 degrees after 18 hours. So what is the reason behind this one? So this is the graph which represents the change in the optical activity, that is mutarotation. First, it shows that alpha D glucopyranose. When the alpha D glucopyranose is dissolved in water, change in the optical activity getting decreased and reaches to a constant value plus 52 point. Then similarly, beta sugar increases and reaches to a constant value. Next, what is the reason behind this one? In order to understand this concept, we should know the anomers. Glucose exhibits the two forms alpha D glucopyranose and beta D glucopyranose, and these two forms are all interconvertible in the aqueous solution through a aldehyde form, that is open chain aldehyde form. Okay, so initially when we place alpha D glucopyranose, that gradually converted into beta form through open chain form, and similarly when we take beta form in water, that will gradually converted into alpha form through aldehyde form and reaches a equilibrium mixer. That equilibrium mixer will maintain constant activity after a period of time. Thank you.